Cell cycle control system. This would be the first part of cell cycle control system. And in this session, you would get an overview of the cell cycle control. So we all know that the cells proliferate or multiply. And every cell, because they ought to multiply, they have what is called as a cell cycle. And within the cell cycle, there are different phases where there are different functions that happen, and all these ultimately help in cell undergoing division. Now, why is the control important? Because one does not want that after cell division, the two daughter cells that are obtained are not having proper genetic information or have damaged genetic information. That is not wanted. So therefore, it has to be regulated. Secondly, if it is uncontrolled, then it can lead to diseases like cancer. So let us look at the learning outcomes of this session. The cell cycle control system coordinates each event. When you say each event, what does it mean? It means that the cell cycle has different phases and every phase is an event. So those events are to be coordinated by the control system. And every event is given sufficient time to ensure that the function has been carried out, and only after the function has been completed that the next event can be triggered. The control system regulates and is also regulated by the processes that are happening in the event. Why is that important? Because that would tell whether the cell cycle has to halt or whether it has to proceed. So if there is something that is not conducive, then there has to be a stop. But if everything is fine, then the cell cycle can move ahead. So therefore, the control system has what is called as biochemical binary switches, which means that they can either get switched on or they can get switched off. And this has to be sensed, that whether it has to be switched on or whether it has to be switched off. There are in fact three checkpoints to regulate the cell cycle progression. One is at the G1 to S junction. There is what is called as a checkpoint, that is called as start, or in animal cells it is called as a restriction point. So that's the first transition checkpoint. Then the second is between G2 phase and the M phase, that is a second transition checkpoint, while the third is within the mitotic stage itself at the metaphase to anaphase stage. So these are the three checkpoints that are regulated. And the cell cycle control is regulated by cyclin dependent kinases and what is called as anaphase promoting complexes or cyclosomes. So these in turn, that is the cyclin dependent kinases and the APCs themselves, are in turn regulated by several other proteins. So it is a kind of, it's kind of a system where there is a hierarchy, and the hierarchy of the control is eventually regulating whether the cell is going to go for division or it is not going to go for division. So we just look at the basic principles of the cell cycle control. So as noted earlier, a cell cycle would always have, or the cell cycle is divided into two major parts. One is what is called as the M phase. So the M phase is where the nuclear division takes place and cytokinesis takes place. So the mitosis takes place in the M phase, and of course the cytokinesis. While the interphase comprises the G1 phase, the S phase, and the G2. The G1 phase is basically where the cell is looking at whether everything is favorable for cell division. And then here at this point, you have a start point. Then S phase is the DNA replication phase, and G2 is a phase which prepares for the M phase per se. So the cell cycle control system is programmed to give sufficient time for each phase. So G1 can be anywhere between 8 to 9 hours. S phase can be anywhere between 10 to 12 hours. G2 can be anywhere between 2 to 3 hours. So each one has been given sufficient time in order for whatever functions have to happen during that time it is being carried out. Suppose a particular function is not over or is not complete, then the cell cannot move from one phase to another phase. That is what is control. That is what is regulation. Now, as mentioned earlier, the cell cycle control system is based on a connected system of biochemical binary switches. So, this in layman's term is that whatever biochemical changes are happening, they tend to either switch on the system or they tend to switch off the system. So, it is only a binary switch. So it is switched on to move to the next phase, or it is switched off if not ready or not needed. So that is something that is controlled. There are three checkpoints at transition phases. As mentioned before, you have what is called as a start transition phase between G1 and S. You have the second transition phase between G2 and M. This is where the cell is going to enter mitosis. So at this checkpoint, the most important question that is asked is the environment favorable for cell division? Why is this question asked at this point? Because once the cell enters the S phase, it is committed to cell division. Okay, if it has entered the S phase and if the DNA has started replicating, then it will go in ultimately for the cell dividing. Suppose the cell is not ready, suppose the environment is not conducive, then from the G1, the cell can go into a quiescent stage or what is called as a G0 stage. Why? Because the environment is not favorable for proliferation. So the cell will remain arrested. It will not go in for proliferation. But when the environment becomes conducive, it can get back to the cell cycle again to proliferate. Then at the second transition point, what is asked is all the DNA replicated? If all the DNA is replicated, then the cell can move forward. So that is a checkpoint. Is the environment favorable? Again, that is something that is to be asked. If the environment is favorable, then again the cell can go to the mitosis. Then within the mitosis, there is the third transition stage, and that is between metaphase to anaphase. During this, the question that is asked is, are all chromosomes attached to the spindle? Why is this important? This is important because only and only if all the chromosomes are attached to the spindle will the chromosomes be pulled towards the two poles, and then the two poles will have equal amount of chromosomes. Otherwise, the purpose of mitosis is lost. The mitosis is basically looking at the fact that if the parent cell is 2n, then after division, the two daughter cells should also be 2n. This is something that has to be maintained, and therefore this question needs to be asked. And when this question is asked, and it is found to be favorable, so there is a tick mark over here and the cell completes the division and goes in for cytokinesis. So this is how you have three checkpoints at transition phases. I am repeating: the start checkpoint or the G1S checkpoint between the G1 and the S phase, the G2M transition checkpoint, which is between G2 phase and M, and the third transition point is between metaphase and anaphase. So in this session we are, or in this particular part, we are going to focus mainly on, the transition at the start point and also the checkpoint at the G2M. Now, there are two key components that are responsible for, the checking at the checkpoints of the G1S transition phase and also the G2M transition. So at the G2M checkpoint, at the G1S checkpoint, there are two key components. One is what is called as cyclin dependent kinases, CDK, and this in itself is dependent on the cyclin. So until cyclin binds to CDK, you do not have the CDK being active, and that is the reason why it is called as cyclin dependent kinase. So this is a regulatory switch. So when you say a regulatory switch, we've already understood that it is a binary switch. So either it is switched on, either it is active or it is inactive. Now, how does CDK itself function, because when CDK has to activate other substrates, what does it do? Being a kinase, it has the onus of phosphorylating its substrates. So when the CDK becomes active, it is able to phosphorylate its substrates, and that phosphorylation of the respective substrate will make the substrate either active or inactive. So different substrates have different conditions. So some substrates, when they are phosphorylated, are functioning or are active, but some substrates, when they are dephosphorylated or when they are phosphorylated, are inactive. So you can have either or. So some substrates, when phosphorylated, become active. Some substrates, when they become phosphorylated, become inactive. Okay, so but the point is that if phosphorylated, the molecules are active. If not phosphorylated, the molecules are not active. Okay, or vice versa. So this is something. So again, here the CDK itself is a binary switch, so it is activating or inactivating a molecule. Okay, now therefore, what could be the scenario? The scenario could be that there may be more than one cyclin dependent kinase. So you may have a cyclin dependent kinase for every phase in the cell cycle. So you may have one for S phase, you may have one for G1, you may have one for G2, that way. But it also may be so that you have one single CDK, okay, for the entire cell cycle, but the cyclin may be different. So the cyclin that is controlling at the S phase is different. The cyclin that is controlling at the M phase is different. That way, you may also have one CDK but several cyclins, or you may have several cyclins and several CDKs as well. So what is important to notice that these CDKs enable cyclical changes in the phosphorylation of molecules involved in cell cycle. So depending on the CDK that gets active, you will have certain molecules getting active in the S phase, or you will have certain molecules getting activated in the G1 phase, or you will have certain molecules getting activated only and only in the G2 phase. So therefore, those that are active in the G2 will now not be active in the S phase. Understand? So there is a cyclical change in the phosphorylation of the molecules that are involved in the cell cycle, and that has been to a great extent attributed to the different cyclins that are active at different stages. Now again, here there is one point that has to be noted, and that is uh, different levels or amounts of cyclins may also uh, be present at different stages. So you also have what is called as transcription regulation. What do we mean by transcription regulation? Well, what is transcription in the first place? When you have a gene getting transcribed to its product, then that is transcription. So from DNA, when you get RNA and that RNA gets into forming a product, then that is what is called as transcription. Now, whether a cyclin is going to be synthesized or it is not going to be synthesized can also be regulated. So what we understand is that the cyclins can undergo a cycle of synthesis and degradation. So the levels of the cyclin itself can be cyclical. Okay, and that's the reason why cyclins are named as cyclins, because in the cell cycle, their amount itself is increasing or decreasing depending on what phase the cell is in. So this is what the understanding is. Like for example, when the cell goes from G to G1 to S, and there is a start point over here, which is the first transition point. So this is the checkpoint. In this place, you will find that the G1S cyclin amount is high. This is the only phase where you see the G1S cyclin high. Otherwise, it plateaus down until the until the cell reaches the G1 again. I hope you've got that. So you see that the concentration of G1S or the activity of G1S is observed only at the start point when it is bound to its CDK. Then when the cell enters the S phase, you can see the S cyclin is active, but the S cyclin is active until the G2M transition phase. And you have then MCDK getting active only when it reaches the G2M phase. So therefore, what is very clear from this is that every cyclin has its activity only for a certain period of time and for a certain phase. So you can see that it is active, it is plateauing down. When it comes back over here, it will again become active only at this phase. Okay, so this is what we understand by cyclical changes in the amount of cyclins. So it will undergo a cycle of synthesis and degradation. To understand it better, you can see over here that for every phase, for example, this is in vertebrates. So cyclin D associates with CDK4 and CDK6 to form what is called as G1 CDK. Similarly, you have CLN3 associating with CDK1 to be G1 CDK. The G1 CDK will help regulate in the G1 phase. Then you have the cyclin E associated with CDK2, that is basically G1S CDK, and that helps at the start checkpoint and is instrumental in commitment to cell division. So once you have this CDK allowing the cell to move to the S phase, then we know that the cell is going all the way to the cell division state. So the cell is going to proliferate. Then you have the SCDK that binds to the the cyclin A that binds to CDK2, CDK1 to form SCDK, and that helps regulate in the S phase. Then you have MCDK helping at the G2M checkpoint, and that regulates till metaphase. After this stage, you don't have the cyclins and CDKs playing the role. Then what plays the role of regulation is the APC, the anaphase promoting complex. So each cyclin not just activates its respective CDK, but it also directs the CDK to a set of substrates for phosphorylation. And that is how it is controlling the events that are happening within a particular phase. So as you can see over here, when the CDK binds to G1S cyclin, it gets active, but after it goes away from the G1S, the G1S start point, the G1S CDK will be degraded. Then what will be formed is the S phase cyclin that will bind to CDK, and once its job is over, that S phase cyclin gets degraded. So the CDK then can bind to the G2M phase cyclin because this is now present. When that binds, it is active, and after its role is over, then the G2M phase CDK is also degraded. So you can see how it is synthesized when required, and after its job, it is degraded. So this is something that it is understood to be happening. In certain cases, the cyclin may not be degraded. They may remain the entire cell cycle, but then they are present in the inactive form. But they get active only in the phase where they have to be functional. So that is also something that has been observed. Now let us understand how the CDK gets active. So we understand that a CDK is inactive when it is not bound to a cyclin. So you look at a CDK that is not bound to a cyclin. So in its active site, it has a binding site for ATP. Okay, why should it have a binding site for ATP? Because the role of CDK is to utilize this ATP and transfer this phosphate to a substrate. So it has to have an ATP binding site, which is present right inside the active site. So therefore, the active site of the CDK will have an ATP binding site, but the active site in the inactive CDK is being covered by a protein that has formed a loop. Okay, so one of the polypeptides of the CDK forms a loop. It is almost like a cave opening being covered by a stone. Okay, now when the cyclin binds to the CDK, you can see how the conformation of the CDK has changed. And with the change in the conformation of the CDK, what happens is that this loop that is there opens up and comes out of the active site. This that is formed is what is called as T loop. Why is this called as a T loop? Because a particular threonine amino acid which was hidden when it was present in this form is now exposed. Okay, now so what has happened is that the cyclin on binding to the CDK, it is active but not completely so. It is still not completely active, it is only partially active. Now, another regulatory enzyme called the CDK activating kinase, CAK, comes and see, it's a kinase, so it will phosphorylate. So it specifically comes and phosphorylates the threonine residue, and once it forms the threonine residue, then the CDK becomes fully active, and now it is going to act as a kinase. So you see how even when the cyclin binds to the CDK, it is not directly converted to a fully active form. It first becomes partially active and then it becomes fully active. Now here you can very well see that the phosphorylation of the threonine residue in this T loop is important for activation. So if the CAK adds the phosphorus, then this becomes active, so it has been switched on. But if something removes this phosphorus, okay, then it will become inactive again or partially active only. Okay, so it is ready to be active but not active. Okay, this is something that has to be noted. Now, in the partially active form or even in the fully active form, the CDK can be inactivated. So we will come to that. So how does CDK get inactivated, or can CDK be inactivated? So what has been observed is that the cyclin CDK with its activating phosphate is in its active phase. But when you have a V1 kinase, okay, which is a inhibitory kinase, this V1 kinase is able to phosphorylate two adjacent amino acids present on the roof of the active site of the CDK. So when you have those two adjacent amino acids getting phosphorylated, then the CDK cyclin is inactive. So although you have the activating phosphate, you have two phosphates which are inhibitory. Can this go back to the active stage? So the answer to that is yes, it can. So yes, it can become active because when you have a CDC25, this is a phosphatase. So it's an enzyme that can remove phosphates. So when the CDC25 removes the two phosphates from the two amino acids on the roof of the CDK, then it will go back to this stage, and this stage we know is an active CDK. So that is how even after the CDK becomes active, if there is a need to control its activity, it can be controlled. So when you want to stop it, V1 kinase can come and add the phosphates, inhibitory phosphates, and it can become inactive. But when again you want to get it active, so then the CDC25 phosphatase has to remove those phosphates to make it active again. So once you have a certain cyclin binding to the CDK, again that cyclin CDK itself can be regulated. This happens with MCDK at the onset of mitosis. This is how MCDK is regulated. Another way of CDK inactivation has been observed. What has been observed is that there is a protein called P27, which is basically a CDK inhibitor, and this P27 can bind to both the CDK as well as to the cyclin. So as the P27 binds to both the CDK and the cyclin, there is a conformational change, very obvious conformational change. And also what is observed is that the P27 gets inserted in the active site in such a way that the ATP binding site is blocked. So with no ATP being present, anyways the CDK cannot transfer a phosphate to its substrate. So therefore, P27 cyclin CDK complex is again inactive. So you can see how An active cyclin CDK complex can be inactivated by binding of the P27. These are nothing but CDK inhibitors. So this is an understanding of how the key regulatory molecule is also under control or can also be controlled. So to get an overview, when the cell is in the G1 phase, and the environment is favorable and there is G1 CDK, G1 CDK will promote the formation of G1S cyclin and S cyclin. G1S cyclin can enable the cell to go from the G1 to the S if everything is fine. Okay, S cyclin will also then proceed to carry out or to help the cell carry out DNA replication. But suppose the cell finds out that its DNA is damaged, then both G1S CDK and SCDK will be inhibited because the cell cannot afford to have its daughter cells after division have DNA that is damaged. That is not, it is detrimental for the daughter cells. So it cannot allow that to happen. So therefore, at this point, you will have, even if you have the G1S CDK and the SCDK form, they can be inhibited and the cell can wait until all DNA has been repaired and then it can move forward for replication. So this SCDK, after it goes from the first transition phase, you will find that MCDK becomes active. The MCDK is at the G2M transition phase, but you can have the MCDK also inhibited if the DNA has not been replicated properly or there are damages that are yet to be repaired. So that's when you can have the MCDK inhibited. It cannot move the cell forward to mitosis. Moreover, we know that DNA replication should happen only once. If the DNA replication has started twice in a cell cycle, again MCDK will not allow the cell to go to mitosis. This is something that is very clear. Then of course the third transition phase does not involve the CDKs, but it involves APC, that is the anaphase promoting complex, and this takes care of, so its synthesis is taken care of or activity is taken care of at the anaphase, metaphase anaphase transition cycle. So therefore, let us look at the conclusions. The cell control system is based on the principle of molecular binary switches that enables spacing out different phases of the cell cycle as well as decide whether the cell should go to a particular stage of the cycle. Sorry for the spelling. Of the control systems, two control molecules are CDKs and cyclins, which in turn are regulated by different signals and molecules. The cyclins are cyclically controlling the different CDKs, which in turn are able to activate or inactivate its substrates by phosphorylation. The CDKs can also be inhibited by phosphatases and CDK inhibitor proteins at different regulatory points. So the cyclin dependent kinases and cyclins can peak up or dip to start a stage and stop a stage in the cell cycle. This control and guarding checkpoints has made these molecules safeguard from diseases like cancer. Thank you.